We've reached our last video for the biochemistry unit, and out of the four macromolecules, proteins have the most varied function and will appear most frequently in the units to come. For now, though, this is video 5.7 to introduce you to proteins. Now, while a lot of what's in this video will follow similar patterns as other macromolecules that we've studied, there are some goals that are unique to proteins over here. Press pause for a closer look. One thing that makes proteins so interesting is all the different shapes they come in. This little guy right here is Rubisco, and not only is that word fun to say, but Rubisco is arguably the most important protein on the planet. Rubisco's job is to fix carbon dioxide in plants during photosynthesis. Hemoglobin, collagen, and growth hormones are other examples of proteins, and you can see the unique, folded up shapes of each. The other three macromolecules had jobs that were relatively straightforward, but proteins do almost everything. I don't want you to write down this whole list, but just listen and appreciate all the things that proteins do. So there's enzyme proteins that speed up chemical reactions, structural proteins like keratin and collagen that make your skin and hair tough, and proteins can bind to and carry other molecules and help molecules pass through the membrane. And this slide is just a sample of the many varied functions of proteins. Proteins are the polymer form, and the monomers that make up proteins are amino acids. Amino acids come in 20 different varieties, but they're easy to recognize if you know what to look for. First, you want to find this carbon right in the middle of everything. This central carbon is also known as the alpha carbon. There are 4 different functional groups attached to the alpha carbon. The easiest group is hydrogen, right? And then you see the amino group in blue on one end and the carboxyl group in green on the other end. And the 4th group is called the R group. Now, there's no R on the periodic table, and the R group varies from amino acid to amino acid. And since there are 20 amino acids, there are 20 different possibilities here for the R group. The other 3 groups, though, are the same in every amino acid. Here are chemical structure diagrams of each of the 20 amino acids. The R groups are highlighted in pink on each amino acid, and you can see that the R groups vary from something as simple as hydrogen, which is the R group of glycine, but the R group can also get as complicated as this big old ring thingy on tryptophan. The cool thing about R groups is that the R group can give amino acids so many different chemical properties. You've got polar, non-polar, charged, uncharged, right? So the versatility of these chemical properties in amino acids is what gives proteins the ability to have so many different functions. Even though there's a lot of variation in amino acids and proteins, both can be easy to identify just by seeing some patterns. First, our sponge elements are carbon, hydrogen, oxygen, and nitrogen. And in a couple of R groups, there's sulfur as well, like methionine right there and cysteine right there. Next, you want to look for those 3 characteristic functional groups around the alpha carbon. You've got in every amino acid, you've got a hydrogen, a carboxyl group, and an amino group around that alpha carbon. Proteins also have pretty recognizable naming rules as well, for most of them. If the name of a molecule ends in IN, that molecule is most likely a protein. And also, if the molecule's name ends in ACE, that means the molecule is an enzyme, which are proteins. Just like other polymers, amino acids are put together into proteins using dehydration synthesis. An H is taken from one amino acid, and an hydroxyl group is taken from the next amino acid. And once this water is pulled out, a peptide bond forms. A peptide bond is the special covalent bond that joins amino acids together. And technically, the chain of amino acids isn't called a protein just yet, but a polypeptide. A polypeptide is just an unfolded chain of amino acids, and once that chain gets folded up, then the polymer can be called a protein. If you look at a protein polymer, it can look really intimidating, but just focus on the backbone of that. See a pattern? NCC, NCC, NCC. This is a tripeptide formed by 3 amino acids, 3 NCCs, linked together. Scientists estimate that humans use between 80,000 and 400,000 different proteins. So how can all these different proteins even be possible? Just like in other situations, living things are very thrifty, very efficient, taking that one amino acid template and turning it into seemingly infinite possibilities. The key to diverse proteins lies in the diversity of the amino acid building blocks, and there are so many different building blocks just because of all of those different R groups. Once those 20 building blocks are made, the amino acids can be put together in lots of different orders. Even a dipeptide with just 2 amino acids has 400 possible combinations, and all it takes is to get to a string of 5 amino acids, a pentapeptide, and that gives over 3 million possibilities. Dang! But remember, once the amino acids are linked together, we can't call it a protein just yet. The polypeptide chain of amino acids needs to be folded the right way to be a protein. But why is this folding so important? Well, most proteins need to be a certain shape in order to get their job done, and a classic illustrative example of this is with enzymes. So enzymes tell us how protein structure matches protein function. Enzymes can speed up chemical reactions because the enzyme protein is folded to have a specially shaped binding site called the active site that fits the chemicals that are involved in this chemical reaction and allows the enzyme to orient the chemicals to speed the reaction up. Protein folding is largely just a function of attraction and repulsion interactions between R groups. Positively charged R groups are attracted to negatively charged R groups. Nonpolar R groups interact with other nonpolar R groups, and so on. And to better understand folding, biochemists break down protein structure and folding into 4 different levels: primary, secondary, tertiary, and quaternary. The primary structure of a protein refers to the amino acids joined together by peptide bonds in the correct order needed. Primary structure doesn't involve any folding, so you see right here the primary structure for lysozyme, an antibacterial enzyme found in tears. Now, these are some different diagram representations of the folded up lysozyme, and this unfolded chain right here represents the primary structure. These specific amino acids in this specific order makes a 129 amino acid chain. That's the protein lysozyme. A secondary structure is sometimes referred to as local folding. With secondary structure, the amino group of one amino acid interacts with the carboxyl group of another amino acid that's relatively close by. The shapes formed by secondary structure are alpha helices that you can see here and blown up over here, these twists right here, and beta sheets shown here and zoomed in here. Look at the dotted lines that show the involvement of hydrogen bonds between these nearby amino acids. The next level of protein structure is tertiary structure, which can be thought of as global folding. And another way to think about it is that secondary structure is folding the primary chain, and then tertiary structure is folding the folds. Tertiary structure involves interactions of the R groups of amino acids and can involve pretty much all the bond types we've talked about so far. Hydrogen bonds, hydrophobic interactions, ionic bonds, and disulfide bonds as well. I like this diagram because it shows an example of each of these bond types. Hydrogen bonds, and we've got these hydrophobic interactions. We can tell that these are hydrophobic because they're just hydrocarbon R groups. And here's a new type of bond, a disulfide bond or a disulfide bridge. A disulfide bond is a covalent bond that's formed between amino acids that have sulfur in their R groups. So cysteine, methionine that we saw before. And here's an ionic bond between a positively charged R group and a negatively charged R group. The final level of protein structure, quaternary structure, only exists in proteins that have multiple subunits. For example, hemoglobin has 4 subunits, 1, 2, 3, 4, that come together to make the final functional protein. Collagen is this thick rope that is made of 3 different subunits that are twisted together to make these really strong fibers. What happens in quaternary structure is that R groups on amino acids of different protein subunits interact with each other, usually through hydrogen bonding, ionic bonds, or hydrophobic interactions. But you can see some disulfide bridges as well. Proteins can actually unfold if the proteins aren't kept in optimal conditions. Protein unfolding is called denaturation. Denaturation is bad for 2 reasons. First, a denatured protein won't have the right shape to do its job anymore. And second, once a protein is denatured, the protein can't renature or refold itself properly. Common causes of protein denaturation include high temperatures and pH extremes, when pH is too high or too low. Interestingly, denaturation involves just breaking the weak bonds. And so what that means is the secondary, tertiary, and quaternary structures are affected by denaturation, but the strong peptide bonds remain, keeping the amino acids together, just not folded in the right shape. So let's recap. Of the 4 macromolecules, proteins have the most diverse structures and functions. Still, though, proteins are recognizable by their NCC backbone, formed when amino acids are joined together to make a polypeptide. To become a protein, the polypeptide still needs to be folded in the right shape. The diversity of possibilities for proteins is mostly due to the 20 different amino acids, each with an R group that determines the chemical properties of the amino acid. When examining protein folding, it's best to break down the structure into primary, secondary, tertiary, and quaternary levels of structure. But all of this folding can be undone by denaturation, the unfolding process that happens when temperatures are too high or pH gets too high or too low.